Welcome to my channel with an interesting practicals which we will discuss today on molecular biology. Experiment name is quantification of DNA by using optical density method. Purpose of experiment. The experiment is important for quantification of DNA in a sample and the purity of DNA in a sample. It tells how much pure DNA is and proteins are present in the DNA or not. Materials required: quartz, cuvettes, spectrophotometer, UV lamp, water, 1XT buffer, double stranded DNA. What is the procedure of this experiment? Let us discuss. First of all, DNA concentrations are obtained by measuring the optical density at 260 nanometer using 1 by 100 dilution in quartz cuvettes and a spectrophotometer with UV lamp turned on. Point 2. In spectrophotometer, a sample is exposed to UV light at 260 nanometer and a photodetector measures the light that passes through the sample. Water or 1XT buffer used as a solvent to suspend the nucleic acid and placed in quartz cuvettes. Example of calculation. Let us calculate these values which we have discussed earlier. A sample of double-stranded DNA was diluted 50 times. The diluted sample gave a reading of 0.65 on spectrophotometer at optical density OD 260 nanometer. To determine the concentration of DNA in original sample, perform the following calculations. Double-stranded DNA concentration is equal to 50 microgram or milliliter multiplied by optical density 260 multiplied by dilution factor. Double-stranded DNA concentration is equal to 50 microgram or milliliter multiplied by 0.65 multiplied by 50 because at 260 optical density, as we have learned earlier, the value is 0.65. And double-stranded DNA concentration is equal to 1.63 milligram per milliliter. Here is the law. We will also discuss this law, Beer-Lambert law. It relates the amount of light absorbed to the concentration of the absorbing molecule. It states that there is a logarithmic dependence between the transmission of light through a substance and the product of absorption coefficient of the substance and the distance the light travels through the material, that is the path length. For 1 centimeter path length, optical density at 260 nanometer equals 1 for the following solution. Now come towards next point, 50 microgram per milliliter solution of double-stranded DNA. The path length defines the distance that the UV light travels through a sample in an analytical cell. For pure DNA, optical density is equal to 260 nanometer and optical density 280. Here we have compared their ratio. Optical density 260 ratio to optical density 280 is equal to 1.8 for pure DNA. Ratio less than this indicates the contamination of the proteins and estimation of the concentration would be inaccurate. Hope so this practical will be interesting for you people. Be connected with me for more amazing practical on molecular biology like this. Have a good day. Subscribe my channel too. Thanks for watching this video.